Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry. Brought to you by Beckman Coulter at Bitesize Bio.
Today on Flow Stars, I'm joined by John Tigges, Principal Associate in Medicine at Harvard Medical School, and we chat about his passion for training the next generation of scientists.
We try to give them things to help their career, not just, hey, come in and you're an extra set of hands.
Why he was actually really great on this episode because of his hobbies, being a DJ and MC.
Also, it may be very comfortable on a microphone or in settings such as this where it's not, oh boy, who's going to see this, what's going on.
The importance of sample preparation in flow cytometry.
This is the ultimate of how everybody puts it out there. The garbage in equals garbage out. And if the prep is not done in a certain way, there's no chance.
And his recent promotion.
I was putting together my Harvard CV recently for a promotion. It's a good time. I will say it. I got it.
Hey.
I'm a principal associate in medicine for Harvard Medical School now.
All in this episode of Flow Stars. Hi, I'm Peter O'Toole from the University of York, and today on Flow Stars, I'm joined by the one and only John Tigges. John, hello, how are you?
How are you, Peter? And thank you for the overwhelming.
You know why that is, don't you, John? Because I believe that you are also a DJ and MC. So I thought beyond just your flow cytometry, I had to introduce you in no other way than to give you the proper MC introduction. So we will get to flow cytometry. John, how did you get into being a DJ and MC though? First of all, we'll start on the personal level.
Okay, so that's maybe not as interesting a story as someone would like it to be. I needed money. So while going to school, one of the gentlemen that I met, he was a friend that I had met through high school, but then we kind of reconnected. As I like to say, I went to college and he visited, and his dad owned the company. So I sat there and he's like, I need help. And I'm like, all right. And I need money. So it was a perfect match because, and then it started off with kind of just doing like the pub kind of things and helping them there and stuff. And then ever since it's been mostly functions and weddings. And I found that, interestingly enough, it was a great opportunity because not only was it fun, I love music, also it made me very comfortable on a microphone or in settings such as this where it's not, oh boy, who's going to see this, what's going on. It was kind of just here it is, here's a microphone in your hand and you've got control. So, our trip.
Right, I'll just say we will get to the science in a moment, but I have to ask as a DJ, what is your banker, the track that you will put on to try and get people onto the dance floor? Because weddings are not necessarily the easiest gigs.
Oh, they are not, and they're hard to figure out because you have such a diversity of age. But it tends to be surprisingly something like if you play some Bruno Mars, and to all feel because it's newer, but it kind of has that disco-y vibe to it. So usually a good start there, or you get a list of some things and you pick out the one that's kind of not going to be over the top, but really gets a good kind of feeling in people. And a lot of times too, they're just anxious because they've been sitting at a table and had dinner and now they're just waiting. So.
Yeah, so I've been to conferences with you, John. I've been to the evenings where it ends up with a DJ and a dance floor. In fact, I remember Leipzig vividly well, and Alfonso, who we know in another episode, has got some good pictures of that. But I remember you did not get up and dance that night.
Yes. Well, I guess I tend to try to be a little more refined amongst colleagues than when I'm out and about regularly. Because even just doing weddings and stuff, I find myself at times, dancing behind my sacred place there. And so I guess this half is all Irish. So
if you're going back, if you're half Irish, where were you? Were you born in the US?
I was born in the U.S.
Yeah. So whereabouts?
Rhode Island. So we are East Coast. Still live there because it is a slightly more fiscal answer to the Boston area. Working in academia and trying to live in Boston is difficult. It's expensive. It's on par with the New Yorks, the Londons, and San Francisco and stuff. So kind of born and raised there, and that's why the accent is this kind of half Massachusetts, half New York. We're sandwiched somewhere in between there. So
I know. So you're at the Beth Israel Deaconess Medical Center, which is part of Harvard. Is that correct?
Correct. We are a Harvard teaching hospital. Look at that.
So how did you get into flow cytometry?
So out of university, I worked for an immunology lab. And so it's called Roger Williams University Hospital, and it's part of Brown University. So I got a position there, and then they said, here's a flow cytometer. It was a fax scan. And anything with electronics and stuff, I guess maybe that's the whole DJ part. The electronics and things were interesting to me. So I said, okay. And they said, do you want to try? Sure. And whether it was a fortuitous thing or not, I was really good at it. So had no idea what I was doing because I wasn't taught. I just sat down and started hitting buttons and became very kind of adept at it. And then next thing I know, they purchased what they call now a legacy MoFlow, and gave me that.
The legacy MoFlow.
Oh, it was like an erector set, right? You could put anything you wanted on that breadboard and just go. So
I was going to ask later on, what is your favorite instrument you've ever had?
I've got to say in between, it was my MoFlow XDP because of the things that we tried on it. I mean, this is stuff, I guess, that would interest you from your microscopy standpoint. We actually placed a chameleon laser on that.
Okay, so that's for the multiphoton, so for far red excitation, or to excite the UV dyes, but using a far red for those who are not with flow cytometry and coherent.
Yes, so we had that, and from Coherent, and Coherent also gave me a laser stack. So we pigtailed in 561, 532, 450. We were playing with a bunch of weird laser lines, 808, I think there was, and then we had that 1080. And the 1080 I loved because it wasn't a simple alignment. See it. So for those who've never played with a chameleon laser or out in the IR range, you just can't see them. So it became very interesting on how we tried to align that. We used a Nikon camera because your little digital cameras, when you take photos, do it in an IR so they can actually pick up the laser beam. So we were aligning through all the focal points and stuff using the camera and kept taking pictures. So it was a very long process, but it was all because we were doing opto injection. So it was trying to kind of get the red blood cells to open, insert stuff inside, and then close them up. So it's because red blood cells tend to have this ability to form after they've been deformed. So that's what we were trying to do. And then we later found out that we were successful. We were able to actually get dye in. It was very bright Lucifer yellow. What a wonderful for a dye. And so we were able to get that into the red blood cells, but we read an article that then said that opto injection with a 1080 laser had 2% success rate. and that if we had just used a violet, we would have a 75% rate. So we were able to actually do something, and I joke with my colleague. I said, yeah, I said we were, I guess we can call ourselves the 1%ers because we at least did it once. But boy, were we barking up the wrong tree.
That's I love the concept though. My background is red blood cell biochemistry, so I quite like the concept of actually punching a hole in it and watching it reseal. But
yes, things with deformability through channels, so you get them to do this up and down channels and watch. And the more rigid they are, obviously during disease states, they won't go through, and they get stuck and stuff. So deformability, their vibrations. He believes their vibrations actually there is them talking to one another. And so what he did is he sent it to somebody to put it to music, so that each of the different vibrations were actually musical notes. So he was trying to get it that, say, when they got more rigid in like a sickle cell, if they could play the Darth Vader entrance theme. Oh, yeah. So this is what we do scientifically. We made it's slightly unorthodox, right? And I may have found my my kindred spirit in this world, because neither one of us thinks in a very straight line. Everything is, the crazier it is, the more likely we are to try it. So
isn't that where you get the eureka moments and the big breakthroughs?
yes, this is definitely where they happen. And if nothing else, it's a lot of fun. You know, I'm not going to, flow cytometry is what we do, but staring at dots all the time is not necessarily, you know, very stimulating over a long period of time. So within a 26-year career, I need something to keep the old mind and the wheels turning. So, you know, discussed that earlier, you know, I forget the little details because we're always looking at the big picture. I said we need to put it on the wall, a little sign that says, you know, two things. It's a go or no go and la putanesca. Just throw everything at it and see kind of what's great, you know, the egg roll principle.
I was just going to ask, when did you use your first flow cytometer?
And let's see, it would be back in 1996.
1996, and you've been practicing it for 20 years?
Now it'll be 26 in mid-May.
I'm just trying to strategically work out your age without actually asking straight out, how old are you? But I guess if you're about 47 anyway.
Yep, I started flow when I was just about to turn 21. And I can honestly say that I've grown up in the cytometry.
And so we're not so dissimilar in age, and I do know that I've gone perfectly grey. You, on the other hand, have got a great reason for why you've not gone grey. You've actually just lost hair on the top of your head. Well, why is that? What is your excuse?
I'd like to say, you know, the classic job stress and stuff like that. But I also believe that the two young women I have in my life. Over the years, they have challenged me and stressed me to some extent, where the gray shows up here. And all chin. This went here. But yes, doing this is my go-to when I'm thinking.
So just rubbing the top of your head.
Yeah, so at some point, I'm pretty sure that I've just rubbed most of the hair out, to the point where it just gave up on me.
And you're blaming your daughters for that?
Some of it, yes. I guess they won't watch this. So
no, I'm now going to make sure they watch or listen to this.
I'm not, you know, unless you do some sort of TikTok dance or you're on Instagram, Peter. I don't know if they'll ever see it. So.
Do you know what? I forgot. What was your favorite dance move on the dance floor? Here's your TikTok moment, John.
It could be my TikTok, you know, like kind of, you know, it's.
How old are your daughters?
They are 17 and 15. I have one off to university next year, and the other one starting her sophomore year of high school.
So what? Okay, so your eldest, what are they off to uni to study? Hopefully.
Psychology and criminal justice.
Ooh.
So
Okay.
she is off to it's Roger Williams University, and they are a law school. So she wants to practice law.
Well, there's a wise lady to start with, because there's bound to be more pay in that than there is in.
And plus, yes, nobody. I don't think my younger one talks about medicine. So as I work for Harvard, she keeps asking me if I can get her into the medical school, and I said very good grades, and yeah, about all. Dad's influence has none as far as Harvard Medical School goes. I said, they let me work here, but even that they consider to be an honor and privilege.
Actually, do you know what? She may want to go to medical school. I would say, certainly the microscopists, there's quite a few medics who left medicine and come back into science research.
Yeah.
No, I would say medics only diagnose and prescribe. They are utterly useless without the technologies such as flow cytometry to analyze and tell you what the results are, and utterly useless without the scientists developing the tools to do that and the medicines to cure that. So arguably, she just needs to go into some sort of life science or technology research if she wants to make a profound difference.
Impact. Well, so I tell her these little stories, and it's funny you say that, because just the other day I was talking with somebody and I was analyzing the results, and I said, we really should perfect this and really go through some more. And so he said, well, my boss, another MD, wants to get this and he needs the results, so he wants to do this. And I said, okay. And he said, it's clinical, so the bar is really low. I just was like, I didn't say anything, left, and my two technicians looked and went, so the clinic is very low on the bar. I go, yeah, I said, he's a lovely man, but maybe not who I'm going to if I have a medical issue after talking with them. So it's funny you say that they, diagnose and prescribe, and that's about it. To sit there and go, well, the science, we're setting the bar low. So, okay. I'm like, well, I don't know how that makes me feel. And should I feel more important or less? I'm not sure. So, it's an interesting concept.
Obviously, we are joking about the quality of MDs here.
Of course.
The skills to diagnose so many different things. But I do think, yeah, and I'm sure they also appreciate, we're all part of the same family.
Okay. In all seriousness, we have some lovely physicians that I work with, and they're doing preclinical work, and just being a part of that is the stuff that makes the job worthwhile. That when someone asks you what you do, and you can actually say, well, I'm working with someone to diagnose, to look at ways to diagnose or treatments for this type disease, whether it's a cancer modality or, like we mentioned, anemia. They do stuff with malarias and all kinds of wonderful stuff. And so it's great to be part of that and be able to put the little Harvard logo next to your name. So
I'm proud of York. But yes, I totally get it. Yes. I can't remember where I was going with my next week. We just asked about your daughters and where they're going. Yes. As a child, what did you want to be?
As a child, I actually wanted to be an architect. As odd as that seems, as a child, I was always, and this is why I love going to Europe, is because there's just that different architectural aspect where, the US is relatively young. And so the architecture is more modernized, and I loved when I went to Budapest and just the architecture there and stuff. So that was where I was going. And my first year of schooling, in college, is what I went for, architectural engineering. And again, it was one of those situations where I was taking biology and chemistry courses as part of my undergrad requirements, and got talked into changing majors by my by a biology professor. said, "You're incredible at this stuff." And I said, "That's great. I don't really care." and it was my third year of differential equations that I decided, and architectural engineering may not be what I was born to do, because I'm like, all this math is really hurting my head. And so I discussed it with those professors again and changed my major.
So differential equations sums up why you didn't want to be an architect. Do you like? Nice play on words there. Ah,
It is. So I differentially equated to a different position in life. And it was funny because it was a professor who was a triple PhD teaching that particular class. And this gentleman was way above my mind capabilities. I wizard of some sort. And it was interesting because when I went back home and said I was changing majors and discussed with my father my issues with the differential equations in this particular professor, my father said, "You've got to be kidding." Still there? So my father actually had him at a different university and struggled immensely and changed his major. So this particular professor was very well adept at destroying the entire Tigges family. So.
Oh wow! So you are answering my questions before I get to my questions, and one of those is, who has been one of the inspirations or most influential on your career to date?
Career-wise, influential. Obviously, I do credit family. My father taught me a lot of good life lessons, if not career lessons per se. His little anecdotes I still use to this day. And so, and then just actually the first and still kind of one of the only in the family to graduate from a university. So that was a huge accomplishment, and I was pushed. And my dad was very much a work-oriented, education-oriented individual. Even though he did not graduate, he was very successful in his career. He worked for a telephone company for 35 years, and moved his way up through the ranks. And then once I got into flow cytometry in general, it was a gentleman by the name of Nikola Kutab, a Cyprian. So my first experience with the Greek culture per se, and he was very influential because he taught me more how to be a good boss than anything else, because he was very, as long as the work got done, he was okay with anything else. It was like he told us from beginning, usually when you get someone starting a position, you don't start off by telling them, "Hey, you want to take a 2-hour lunch? Go for it. You need to run errands, go for it." And I was like, "Oh, okay." Being 21 at the time, and someone telling you, live your life, it was like, wait a second, what? But he was just, "Hey, if you want to come in at 5 a.m. and get everything done and then take off at noon." And so I've kind of taken a lot of that teaching into how I try to, manage the core facility and the people that I had the privilege of mentoring and work with me.
So you had for many years a partner in crime.
Yes.
Right term with Vasilios, yes.
It's my work husband. So the first place that we were at, the Roger Williams that I mentioned, I met him there. Showed up kind of on my doorstep. I joke with him and say, "You were like the baby that was left at the orphanage because you just showed up one day and no one knew that he was coming to work with us." It's an interesting story of he had a relative here that had an illness. So his mother, he was supposed to go back to Greece. He was a podiatrist, and his mother asked him to stay and stay with her and make sure everything was okay. And his his aunt, that particular aunt, was very friendly with the president of the hospital. So the president of the hospital gave him a job that even my manager did not know was a position. So, and then we worked together. I moved to Beth Israel here, and he was in between what he was going to be doing next. I was talking with my wife via Facebook, and I said, I'm looking for somebody. So we came back, and so total, I think we worked together like 18 years. And it was weird because when he finally moved on to industry and doing some clinical work for Beckman Coulter, there was many a person, such as yourself, that felt and said it was like, what could be sacred anymore if we broke up? So, the worries of, oh my, work relationships. You guys were the ones that everybody else felt. Now it's like Peter and Karen, right? I can't imagine ever hearing a time of Peter without Karen or Karen without Peter. That was how we were. Obviously, you were still friends, very close. He's my younger daughter, Kylie's godfather. So, we're family. So that's not going to change.
No, say hello to him when you.
Definitely.
Next
time. His daughter was at my house yesterday. So hanging with my wife.
That's very cool. I'm going to change that question. I'll come back to that one in a minute. Nano Cytometry. So actually, I look, I don't do a lot of background research. I shouldn't need to do background research on you, John. I know you fairly well, but actually in your description is one of your areas of development of expertise is nano cytometry. What on earth is nano cytometry?
I'd like to joke and say it's really small cytometers.
I just thought it was something off Mork and Mindy. Yeah, nano. Yeah,
nano, yes. Yes. Robin Williams is one of my favorites. So great reference. Wow. I don't even know where you possibly pull it. That's coming from the stratosphere there. So no, in all seriousness, we got into nanocytometry about 8 years ago. Someone approached us and we had no idea. So this is looking at things that are below the 1 micron in size. It can be known as extracellular vesicles. They have names. The whole nomenclature was debated for years and still is. What should they be called? So, and it's just being able to look at these things. And let me just tell you that there are many a time that I want to go back to just plain old cell biology. Because when you start looking that low, as you probably know, when you decrease a threshold that much, you see everything. And your job becomes getting rid of all the extraneous materials. And that can be simply, you find out that people have contaminated, HBSS or any, PBS. I found out our Millipore water is pretty dirty considering, everything else. And so I think it's a wonderful, wonderful science. It has brought a lot of recognition to my core, has given me lots of speaking engagements. I would also say that most of us probably don't know 80% of what we should in order to do this correctly. Standardization in flow cytometry itself is problematic, that whole rigor and reproducibility. It is a nightmare to try to do this with, instruments that were not built for this. We're trying to use a hammer to do everything, to put in the screws.
You say instruments aren't built for it. Light
is not built for it.
Yes. Isn't built for cytometry.
I became extremely doing dark field microscopy. So dark field is one of those things that actually kind of works with these guys. When everything else is dark, you can see the little guys that are the only light. And so it's, for For those who are not familiar with dark field, I always think about it like Starry Night, right? When everything, there's no clouds, the moon is completely blocked, and you've just got those black nights where you can see the stars perfectly, and there's the Big Dipper and Orion. It works the same type way. And my microscopy guru, that's how he always describes it to us. He's like, it's like the Starry Night, and it's interesting. I'll ask you, this isn't supposed to be about you, but are you also big into astronomy? A lot of the microscopists I know love astronomy and the telescopes. And...
Not massively into astronomy. Certainly, I know my constellations, and I'll do a bit of stargazing occasionally. Bizarrely, Chris Lintop is we have a program called Sky at Night in the UK. And the presenter of that is Chris Lintop, who's actually also a guest of the podcast series called The Microscopist with us. And this is the crossover between the astronomy and the sciences. And to actually understand the universe better, he started something called Zooniverse, where you use citizen science to start helping analyze all this data. Mine's loads of data, and a computer can't do it. You need to train the computer, so you need the human input. So he's got thousands, probably hundreds of thousands of volunteers doing this. But of course, microscopy needs the same help. And so then Zooniverse now has a project for essentially called Extracellular with Lucy Collington, Chris Petty, Mark down in London, that do just this. And there are loads of volunteers out there helping solve cancer by just literally drawing round images, because a computer cannot do it as accurately as a human. So no, not astronomy a little bit, but it certainly has those relations through, and maybe bird watching is more my thing. So I still have the telescope that I can use for both.
Okay, because one of our big things eventually is to take a pilgrimage to one of these, super telescopes, whether in Africa or I think there's one in Peru. So the two of us are like, one day, this is where we need to go. So it's interesting how all these things wrap in, like you said, tie into one another, and how just the interest in optics itself spans so much, and just, and how it gets the juices flowing. I mean, my table's usually cluttered. They cleaned it behind me for today. It's usually cluttered with photonics magazines and spectra and all these little fun things that I think when I'm on the train reading, the individuals are like, what is going on here? Because it's just, I don't know. I guess certain things nerd me out. I realized that this passion when Cyto was in Edinburgh, but I went over to Glasgow where the coherent factory is.
Yep.
And when I actually came back from it, I called the family and was talking to my wife and my two daughters, and was like, oh my God, I went to the laser manufacturing facility for coherent, and they had all these lasers there, and they were talking about, how they can manipulate, the neurons and all this fun stuff within the mice and the brain, and how these technologies can really, with Parkinson's and all this. And I was just going off for like about a 10-minute monologue. And at the end they went, huh? And I'm like, oh man, I just kind of nerded out. I said, I got to realize this isn't your Disneyland. This is my Disneyland. And they're like, yeah, because I was like, you need to come and you need to see this stuff. And they're like, doesn't sound like a very fun vacation.
No, but very much a busman's holiday. I think if you call it in the UK, where you go off and do a similar job but on your holiday when you go overseas. I liked your analogy to astronomy, actually, because even going back to Nano Cytometry, I used the analogy of the stars, of signal to noise. You can see very few stars when there's lots of light being scattered in the sky. In fact, you can't see a single star in daylight just because it's the light scattering. But your fluorescence is still shining, or in the astronomer's case, the star is still shining. Same amount of light, it's not changed, you just cannot see it over that background. But for nanocytometry, it's all the other particles that are getting in the way of those really faint stars that are interfering with the image. And this is what scares me about nanocytometry. This is what I hate, the fact we can get so small, and for bacteria and some EVs, we can see them, but there's always going to be particles. It's not black and white. It's not that all of this size. There's a slope into the noise, and we can't see them all. And I really struggle to cope with knowing I can't see everything on my cytometer or microscope, whichever way it is.
Yeah, I think that is the most frustrating part, and I'm sure that seriously has contributed to some of that hair loss we spoke of, is just trying to figure out, always trying to figure out, how can I really get that signal to noise to be better? How can I eliminate more dark noise? And this is the ultimate of how everybody puts it out there. The garbage in equals garbage out. And if the prep is not done in a certain way, there's no chance. I mean, there's some kind of, okay, let me see if I can save your sample when you're talking about cellular biology. There's nothing. I mean, if you are not, I like to tell people, I said, you're trying to get donors, right? But you're really worried about, hey man, what did you eat today? Have you had a latte? Your lipoproteins, like your apolipoproteins, that you can see cloudy layer that just sits there. And then once you get rid of that, you're talking about chylomicrons, which span the entire size range of extracellular vesicles. And again, more lipoproteins, and you can generate micelles. And so it's just horrible kind of thing. And companies are on this kind of arms race of who can put out these products. And so size exclusion chromatography has become this kind of go-to thing, where ultracentrifugation was a huge thing, right? And then people came about and said, no, there's problems there. And then size exclusion became a thing, and it was like, well, there's problems there. And then it was like, well, what about, flow fractionation and stuff? Well, there's some problems. So now you read papers that are like, okay, we did 3 processes.
Yeah.
And like you said, now I can kind of see things and I know it's clean, but I lost 80% of anything that could possibly be useful. And so every single, I don't know how many of these nano talks that you may have attended or on a webinar, almost everyone in some slide has the iceberg. And we can only see the tip of the iceberg. And so I like to think that we're getting a little bit lower and we're starting to see things that are more interesting. And I like to think that some of our work has been instrumental to that. Why not? Why can't I think that? Doing science.
Oh, I've just had a brainwave. But do you know what? I'll talk to you when we finish that, John, on that one, because we'll keep that one till afterwards. I've just had a thought on that. I think what's really, nanocytometry, and it's not just nanocytometry that struggles. It shows that technology has evolved really rapidly over the last 20 years. And there's still such a long way to go. There's so much still to develop instrumentation-wise, probe-wise, and to understand, so research-wise. And actually anyone listening who's a new scientist coming in and wondering where careers are, wow, there's so much still to be learned and developed in this area. I think 10 years ago, I was thinking cytometry may burn out. Other technologies were coming to the fore. And oh my goodness, it didn't burn out. It kind of just had a little plateau, and then it's rocketed again. And if you look back in time, it took off from the zero, and it started with a plateau and then went up, then plateaued, then went up and plateaued. And it's up. And the gaps in the market mean there's still plenty of rapid rises still to come. So we're good until our pensions.
I think there's two, when I look at it, I think it's twofold. You could, I think one is we're very good salespeople. So while flow, like you say, there was this level, is we started getting the immunologists to be like, hey, without us, you can't figure anything out. And so we need you to push the companies to start making ways to look at multitude of things simultaneously. So 30, 40 colors. and that kind of trend went. And then the flow cytometrist said, oh, it's great. You can do Western blotting and some PCRs, but even with your little extracellular vesicles, now, if you want a multiplex, you need us. And so we're very good at adapting, selling our skill set, and kind of self-promoting. So we're important. It's here because, like you said, there's a lot of things that can be done computationally, and a very good mathematician can take a lot of the burden off of analysis and what we're trying to do. And I know I've gotten to be very good friends with a lot of mathematicians, people that are whizzes at computational analysis, and that's where things have gone. And then the second portion of that is I say to people, we've had a lot of new developments in flow cytometry. I'm waiting for that next novel thing. I kind of caveat that by saying new means that it's just the technology and we're using it for a different purpose. Novel, turning the flow kind of world upside down, and things that were done between the flow part and just cytometry when it was invented, just looking at air and air, same principles, but there was no fluid to it whatsoever. It was looking at for aerosolization and things of that nature. So I'm like, what would be that next kind of over? Do we lose the fluids again? The lasers become some sort of other thing. And that's why these conversations with guys like Bill Telford, who are laser gurus, are so fun because of the thought process. And even looking at, hey, one really kicked up white laser and lots of pinwheel fun, or prisms. And, I like to say flow is about smoke and mirrors. So
yeah, I can't see us putting our electron beam through the flow cytometer just yet, but never say never. Never say never.
Say never.
Changing tack a little bit, what has been the most challenging or difficult time you've had in your career to date? I say to date doesn't mean you're going to get more difficult times coming forward.
Something I didn't. At 11 o'clock, someone will be giving you a little piece of pink paper and a cardboard box to put your stuff in. No, seriously, I think the most difficult aspect, and I think a lot of people have the same thing, and we mentioned it, is the revolving door. It's great to meet a bunch of people, but I'm the kind of person that, I put a lot of stock in relationships and put a lot of stock into, meeting people and learning about them and understanding their goals and stuff. And so when they leave, it's almost, over the years, it's like little pieces of you are taken with them. And so whether it be someone that's worked with me directly within the Corps, I was putting together my Harvard CV recently for a promotion. It's a good time. I will say it. I got it.
Hey.
I'm a principal associate in medicine for Harvard Medical School now.
Congratulations.
Yeah, thank you. And part of that was a list of mentees. That was a huge thing they wanted to see. And just looking at that list, it kind of hit me a little. Wow. Look at all the individuals. And it wasn't so much like, look at all the individuals that I've had some influence on. It was more, look at all the individuals that have been part of my life and now have moved on. And that's difficult. That is a very difficult part, just seeing them all go over time. And that's the nature of the beast. I get it. But, some have come back. It's been great. Seeing them again in different positions and being able to interact with them in a different level. But, we said we'd talk about it later, but, individuals that you've sent along the way from York, like this gentleman, Andy Stone, Harry Potter lookalike contest winner. He loves that one. But he embraces it. He dresses up like that for Halloween, he said. But individuals like that.
That's not dressing up, is it, if he already looks like it?
No, I guess it's not. I guess it's just putting on a scarf at some point. But he's such a wonderful individual, and I use that as an example that eventually he will come back to York. He wants to go back to, to England and the UK. And so just the 6 months that I've spent working on nano cytometry with him and just getting to know him is unbelievable. I actually have a Christmas card sitting above here of Andy and his significant other and his kitty. So you make relationships with these individuals, and it just becomes more and more difficult to lose them.
And on that, hey, Andy, if you're listening or watching this, where's my Christmas card? Just out of interest. Yeah,
yes.
You'll be back to York at some point, so mate, get that card sent.
He said it was in the mail. Hold on. Let me see. He wants to know if you moved. He thinks that may have run the problem.
Thinking about those as some of the more challenging times, it's sort of losing a bit of you each time they go. Do you feel the same about, we've talked about this in the past, it's quite interesting to hear your views on this, about your attachment to the facility itself as well?
Yeah, so I think that's it. We've started talking about this in the past, and I'm sure others have the same. When I came to Beth Israel, we were a fax scan and an FC 500 facility with a one employee for the flow core, the guy they hired, and hundreds of users for 2 instruments. And so to this day, we've moved to a new building about 10 years ago, built a wonderful facility. We now have it's myself, and we have employees. There's 1 open position still, and I have 3 interns all working in the core on projects.
So, John, how many staff did you say you have currently working?
2.
Say again. Sorry, you broke up there.
Oh, sorry. We have 2 employees, 2 technicians, 1 open position, so there should be 3, and then but there are 3 interns from a local university.
So you're about 6 staff currently working in the lab.
Interns, though, tend to be more, they learn the flow cytometry, they do work along with the researchers, but they have projects that we give them to assist on. And it's great because they love it. We have a young lady who's leaving us in May. She's been with us for 2 years as an intern. She's getting a job in industry, but she will leave with being first author on 2 papers and a co-author on another paper.
Wow.
So we try to give them things to help their career, not just, hey, come in and you're an extra set of hands. And so that's great. But again, even with them, it's like, because of their age and my age now, it's like children, right? It's like watching your children. And while, like we said, it hurts and it's a little piece of you that goes, there's also a sense of pride, and that feeling that they got something from you and will take that along in their careers. So it's interesting. There's, as you said, dynamics, and the same thing. If I ever decide to leave this facility for, I don't know, what might be greener pastures, or someone finally convinces me that the industry is not the dark side, and then I would love to see someone here who I feel is just as dedicated, because again, the facility itself feels like a child. Saw it and brought it from a baby right up into its now adulthood, and that they can maintain the quality and even hopefully, as you know, and we've discussed, it's not just about status quo. It's the next person who can push it to a higher level. You always want to see the people that you mentor go beyond. Maybe it's just me, but I always want to see them better than I am. I have no problem with that. I don't have an ego in that sense of, oh my, someone I trained is better than me. No, I think if they're not, I haven't done my job. And so I see my job as an educator and a mentor, not just the guy who looks at some instruments and they laugh at when I take them apart.
So I'm gonna think about this. You've got all this time. Some quick fire questions for you.
Okay.
Early bird or night owl?
Night owl.
DJ. obvious. Neat or messy?
Organized confusion.
Organized confusion. Okay. PC or Mac?
Mac.
Oh, McDonald's or Burger King? Burger King?
Yes.
No.
They give out little paper crowns.
That's I'm not going to go there. Tea or coffee?
Coffee.
Coffee. Wine or beer?
Beer.
Beer. Okay. Chocolate or cheese? So, give us an before it, John. Just keep breaking up there. Chocolate or cheese?
Cheese.
Cheese. So sweet or savory? I'm going to guess savory.
Savory.
Definitely. Okay. TV or book? Was that a book?
Yes, a book. I like.
So what are you reading at the moment?
So I'm waiting upon reading once my my technician Brandy is done with the Witcher series. So I have watched it, but I have not read the books. So previously for me was any crime novel. Some of them being so I guess whether it's good or not, I have this infatuation with the Criminal Mind or your serial killers. And so sitting there going, I don't know if it's just the biology aspects, but I'd love to see kind of brain pulses and scans of these individuals. So, and some of the books have it in it. So it's very interesting. It's intriguing.
So that's where your daughter gets it from, I guess, then.
Yes, they have. So my younger daughter is actually, she likes watching, but she's read like the whole Harry Potter series like 3 times. And she actually, from last time I was in Dublin and I had the kids with me, she actually bought a book on the famous Irish criminals and from like Kilmarnock jail. So, which was very intriguing to me. So, and it was, she bought it with money that she won off of an individual by the name of Tidar.
Okay.
Winning money from Tidar in a pub was not difficult.
Especially no, no. I can imagine it's not so difficult.
Yes.
No offense, Tidar. Obviously.
No offense whatsoever. It was because of his language. He promised her he would not use foul language, and if he did, he would give her money. Well, let's just say he lost that, and she made out like she robbed a bank. So,
Was that for every word? Is that why she actually robbed a bank at the end? She had that amount of money.
She got that kind of amount of money. So yeah, it was, and she couldn't have been happier. To this day she loves him.
Oh, he's a great guy, though.
Oh yes.
What's your favorite film?
Day Off.
Ferris Bueller's Day Off. Utter cult.
That's one of my favorite films. Along with every Halloween, I watch The Crow with Brandon Lee, the original.
Okay. So you got your Halloween movie. What's your Christmas movie then?
The Christmas movie would be National Lampoon's Christmas Vacation.
Oh, Cherry. Yeah, we do watch that every year. We've got a few we watch every year with the family. But yes, that is one of them.
And of course, so we go to my wife's cousin house and we tend to watch Die Hard as a Christmas favorite. For some reason, I don't know how that became a Christmas movie.
Come on, it's a Christmas movie, isn't it?
Yes, it is it may be the ultimate Christmas movie.
It's excellent. What is your pet hate? What do you really dislike people doing or things in life or something that's happening? What is your pet hate? Start with an because you're just breaking up at the start of the sentences.
Yes. My pet peeve. Full sense of entitlement. I guess it's because I feel like I've worked hard to earn where I am. And so those that just show up and believe everything should be given to them really gets me going. I mean, you may have certain opportunities, but then it's on you to make it happen. Yeah, that's kind of a pet peeve.
Yeah, that's a good one. I can see that. If you support a football team that I support, I think a lot of our players seem to have that sense of entitlement that really they are not delivering. What about, do you have any bad habits?
Bad habits is I get that whole distraction thing very easily. I tend to be thinking of 10 to 15 things at all times, and this sometimes drives individuals that I need to get things accomplished crazy, because they're like, well, you just told me to do. Yeah, forget that for the moment. And I just thought of this one. And so that's a really bad habit. And then my wife hates the fact that when I get nervous or a little bit upset, I chew the inside of my lip. And so she knows and says, she'll look at me and be like, if you're angry, just say so. I'm like, I'm fine. She's like, well, then why are you chewing your lip? It's a tell, I guess. It may not be a bad habit. It might just be, if you play poker with me, look for the chewing of the lip.
I was just thinking of that. When you've gambled on the wrong hand, just look for that lip
Yes.
chewing. But now we know that you could be bluffing.
Yes, there we go. Maybe I just put that out there.
That's not so good at that point. We are nearly out of time, and I've got a few more questions I really want to ask you. First of all, what do you do to chill out after a difficult day, after rubbing off all the top of your hair that you keep doing with your daughters or with work and losing your mentees, as it were? What do you do to chill out at home?
Chill out for me is hitting the gym. I get to work off all the excess energy and stress. So my younger daughter, she has become a gym rat. So even when I think I don't want to go, I go. So she does that. And then I love to play basketball. I've been playing since I hadn't picked up a basketball until I was like 14. And it was just on someone needed an extra warm body. And then I just because I was always a football player. I was playing soccer here in the States, and this is why I still like it, and people think that's weird for an American. I'm like, yeah, but I played the sport. But then I started playing basketball, and it was a lot easier to find, a couple of guys to play basketball than it was to find a few people to go and just to pick up soccer games. So
soccer's still better. Sorry, that's a personal opinion. You do work now. We started to branch out. We've talked about teaching, mentoring, but you're also thinking community-wise, networking and being responsible. So you're now doing work with the ABRF. So do you want to describe what the ABRF is? I know what we have, the CTLS, and actually I've been part of the international committee for ABRF as well. But you're more involved with that now. So go on, just introduce ABRF and what you're doing at the moment.
Okay, so the ABRF is the American Biomolecular Resource Federation. Very fancy term, I guess, which is a group that kind of, as you know, from your experiences, cores in general. So for management and all the particular cores. And I belong to the flow cytometry research group of individuals that we put projects together. So same thing as type writing grants. You write, a grant to the society to see about, what you'll actually fund with papers in mind, manuscripts. So the thing I'm involved with them right now in is looking at a technology and technical pathway to a core facility. So working with local community colleges or, universities to set up a program so that individuals aren't just coming to a core saying, oh, well, I did the biology requirements. It's going to a core having core skills. And that entails, which I worked with the group to say, well, we need to add in there the management portion. So that's what we were saying. The individual who not only knows the technology, but knows the people skills, knows the salesmanship, knows how networking and company relations can benefit, and understanding budgets. And, why can't I hire 6 people to work in the core? We'd be more efficient. Well, the budget says, no, you will not. So, and also understanding how to work that budget to get what you actually need. So it's a wonderful kind of thing that I'm happy to be, involved in. And also the group is working on how to get people to actually do titrations. And also what happens if you're sorting larger cells on your normal sorter. So, people don't, they say which I agree with 100%. People don't take in consideration that you're using a 3 micron bead to set up any kind of drop delay. But what happens when you start shoving things the size of my head through a nozzle, even a larger nozzle? How does that affect side streams and everything else? So yeah, we use that analogy all the time. The size of my head and the brightness. I can describe nano cytometry by how the light hits off the top of my head. So
that's like, look, you've got 2 laser lights going on as well. So you've got two.
Yes, we have 2 laser lights, so it's good. I've got side scatter and back scatter going on. So
I just going back to the finances side. So many core staff don't like the finances. Ultimately, it's what empowers us. I actually think it's yes, it's a pain. Yes, it's difficult. Yes, it feels awkward. But I do think when finances are put properly into place, it can make a profound difference to how a facility can really operate, because you can justify, users are justifying it. You don't have to have it because people say, oh, yes, we want it. People are really putting their money, it's voting with their money if they need that facility, they need that staff. It's tremendously empowering. I think for the ABRF, just like CTLS, and actually even with the Royal Microscopical Society, we even teach courses on facility management, for people wanting to go into this sort of career path. Because
I saw that on LinkedIn for CTLS, that they have an actual couple of day intensive course on the business portions of a core. And I've always been interested in that. Like I said, when we started, I grew up in this. So for better or for worse, I've learned business administration. I've learned legal studies there because of contracts and just how things behind the scenes need to operate in order for a facility, i.e. a small business, to operate effectively. So I think that's what's get lost sometimes in the research portion, is that we are a small business. That mom and pop store in the mall. You've got the big entities of the clinical and things that are bringing money to the hospital, and you've got your little portion of it as people are walking through, and you're trying to get them to come into your little shop and stay relevant and financially sound. So it's an interesting, I always tell people that, and they go, that's an interesting way to look at things. And I'm like, well, we are. If we don't have butts in seats, we're going to fail. And so you need to figure out how can you build those relationships. And the other thing that has been relevant on the Purdue side even lately, and I've said it from beginning of time when I started this job, we are the bartenders of the science world. I know things about people that maybe I should not know, but here I am. And I've been asked for advice on so many different topics, that we could do an entire other session just on us going back and forth on what's the strangest thing you've ever been asked.
Yeah, the bartender, the agony aunt, or
yes.
Very much so. John, we are up to the hour. So I'm going to say, actually, I don't know, you said the bartender, but I'd also say through listening to this and the diversity of your career, your job is, I've also got the teacher, the businessman, the legal man, the accountant, the DJ stroke MC, the family man, but maybe most importantly, the scientist that's there. John, it's been great catching up with you today. It really has.
Great, and I can't wait to actually be able to see you in person.
Wow.
So yes. We can share some more stories and a pint together.
No, there's no point coming over on the RMS facilities meeting because you know it already. We need to think of another event. We'll hook up soon.
Yes.
But for those that are watching the Flow Stars, do go back, watch the other ones in the other series that are out there as well. There's some great speakers, including Alfonso, who you've heard a lot about today. Don't forget forget to subscribe to the channels. And John, as always, utterly brilliant. Thank you.
Thank you too, Peter. It was an honor.